Imagine this. Old monkeys get human genetically enhanced cells for almost a year, and their biological age moves backwards in many parts of the body, like brains, muscles, skin, immune system. And the craziest part, during the observation time, researchers reported no serious side effects, even though these were human cells inside primates. This is not a mouse study anymore. This is a very close to human. So what exactly did they inject? The key word is FOXO3, what this Chinese rejuvenation tech is. This study is about a cell therapy called SRC, senescence-resistant mesenchymal progenitor cells. In simple words, these are repair cells made to be resistant to aging, so they don't break down in an old body. And the scientists boosted them using one of the most famous longevity genes, FOXO3. So these are not regular stem cells. These are cells engineered to stay strong under stress. But what is FOXO3 really? Magic or real mechanism? Let me explain in the simplest way. FOXO3 is a transcription factor, basically a master switch. It turns on cell's protection program, stress resistance, damage control, repair, and cleaning up cell junk. As we age, these protective programs often get weaker, inflammation rises, damage builds up, tissues decline. In this technology, FOXO3 was boosted so the cells stay longer in protection mode. But theory is easy. Real results matter, and that's where the numbers get wild, and we will show them. So the researchers didn't only look at how the monkey feel. They used aging clocks. One based on gene activity, so-called transcriptAge, and one based on DNA methylation. In the summary of the results, SRC therapy reduced biological age by 3.3 years on average, and it affected about 54% of tissue tested. Regular wild-type stem cells also helped, but less, around 2.8 years in average, and about 31% of tissues. So it's not just stem cell help. The enhanced version had a bigger and wider effect. But which tissues changed the most? And this is where the story gets interesting. The strongest age drop was reported in skin, which was about minus 5.5 years, in lungs, in skeletal muscles, in spleen, in hippocampus, it's a memory area, and the DNA methylation clock showed similar direction of change in brain and other tissues. This matters because these are not cosmetic tissues. They shape real life, breathing, immunity, movement, and memory. And since we mentioned memory, let's talk about the brain. In overview, the SRC monkeys did better on memory tests compared to controls. MRI results were described as showing a more youthful brain structure in some areas and better connectivity in memory-related regions. The hippocampus is one of the most vulnerable brain regions in aging, so even a small shift is a big deal. At this point, it started to look like a systemic change, not a one random marker. But the brain is the headline. The real foundation of aging is bones and muscles, and there are results there too. The summary reports less age-related bone loss and better bone structure in SRC group, plus improved bone markers. In muscles, the biological age decrease was stronger with SRC than with controls. In real-life term, that means less weakness, fewer falls, fewer fractures, and more independence. So how can one therapy affect half of the body? And does the cell stay forever? Here comes the most interesting part, the messenger system behind the effect. A big mechanism candidate is exosomes, tiny packages that cells release. The idea is SRC cells may work not only by becoming tissue, but by sending signals. These exosomes may reduce inflammation markers, reduce damage signals, and reset gene programs in immune cells. If this is true, the future might not be cell injections, but signal injections. But there's a twist that makes this story powerful and expensive. They did infusions every 2 weeks for 44 weeks. In the popular summary, the dose is described as 2 million cells per kilogram per infusion. So it's more like a long protocol, not a one-time miracle injection. And weirdly, that makes it more believable. They tested something closer to a real therapy schedule. Now the nature question is, while this is not available to everyone yet, can we activate similar cell protection programs in other ways? Here is what matters in Chinese therapy. They improved cell resilience, largely connected to FOXO3, and they got systemic effect. Now let's connect this to something more accessible. Can we support some of these protective gene programs without cell therapy? I have a document I can show on screen, a Macrolife Biotech report. It's Germany 2025. It is an in vitro study on primary human macrophages. They treated the cells with synthesized iron with our product for 24 hours and measured gene expression. And in their longevity panel, the numbers are very clear. HMOX1 plus 100%, FOXO3 plus 75%, NRF2 plus 88%, etc., etc., etc. In simple words, these are gene programs linked to antioxidant defense and stress resistance, things that show up again and again in longevity science. Of course, this is not the same as injecting engineered cells into primates. That is a different level, but the logic overlaps. FOXO3 and NRF2 are about resilience and protection, and SIRT1 is often discussed as key node in metabolic adaptation. Now let's get back to the main question. Why does this Chinese technology look like a start of a new era? During the 44-week period, the summary reports no major red flags like tumors or strong immune rejection signals. Of course, long-term follow-up for years is still needed for any gene-edited cell therapy. But the fact that primates received repeated human cells infusion for almost a year with no serious issues reported in that time window is a big signal. So what does it mean for humans and the dream of real age reversal? And also, why this feels like an engineering, not a miracle? The real breakthrough is not a headline, monkeys got younger. The real breakthrough is in primates, aging markers can move in a younger direction systemically, not only disease by disease. Today it's a complex lab protocol with many infusions. Tomorrow it may become standardized, and later it might become a combination of cells and their signals like exosomes. If very long life used to sound like philosophy, now it starts to look like an engineering problem. Thank you everybody, comment, subscribe, and goodbye.